myself Ankit. Hope all of you are doing well. So we were looking at the chapter called Level of Organizations of Life, and we today in this part we will be looking at the overview of the cell. So without any further ado, let's get started. So this is here I have drawn a typical animal cell, and here I have drawn the typical plant cell. So we will be describing the whole structure of the cell, that is what are the organelles, how the cell is made of, each and every details will be covered in this video. So to start the video, I am introducing you a concept that is called cell theory. So what is cell theory? Now two scientists, Sladen and Swan, gave the idea of the cell theory. So what is cell theory? Cell theory is what they have said, that every cell or each cell is the structural and functional unit of life. So structural and functional unit of life, and each cell have originated from its previous cell. This is, I can call the parent cell, and this is a filial cell or F cell, just naming them. So according to the cell theory, the scientists said that each and every organisms that we can see in our daily life have a structural and functional unit, that is the cell itself, and all the cells have originated from the parent cell. So this is the cell theory that was described by two scientists called Sladen and Swan. And the next part that I am going to tell is the cell is the basic unit of life. So এই উত্তরটা আগের প্রশ্নটার মধ্যেই লুকিয়ে রয়েছে, যেটা মানে কি, যে একটা সেল, when we are considering a life, যখন আমরা কোনো একটা অর্গানিজমকে নিয়ে ভাবছি, so organism is basically a result of many cells, many cells. So how it is? So let me write. Cells usually made or form tissues. Tissues usually forms organs, and many organs forms the whole body. So the idea is that cell is the basic unit of life. Yes, it is the basic unit of life because the whole life or the whole body of the organisms is made up of many cells which have converted into tissues, and with tissues they have converted into various organs. So this is the introductory part of cells. So there are two types of cells from the perspective of evolution. So evolution এর দিক থেকে যদি আমরা খেয়াল করি, তো আমরা একটা সেলকে দুভাবে ক্যাটাগরাইজ করতে পারি, ঠিক আছে? সো এভলিউশনের দিক থেকে আমরা একটা সেলকে দুভাবে ক্যাটাগরাইজ করতে পারি। কি কি না, আমরা যে সেলগুলো, the cells which have come earlier in the process of evolutions can be called as prokaryotic cell. Prokaryotic cells, and the cells that gradually evolved from the prokaryotic cells to a newer version of cells are called eukaryotic cells. So these are just old cells and new cells according to the evolutions. Okay, so basically there will be some distinct features between them. As they are old, they have some primitive characterizations. As they are new, they have some evolved characterizations, which we will be dealing in the next part. Okay. So I have categorized a cell. I have told that there are prokaryotic cells and eukaryotic cells. So what I have drawn here is the structure of an animal cell. Okay, this is the perspective called organisms. I am differentiating, or I am making difference of cells basis of organism. So there will be animal cells, and there will be plant cells. So স্বাভাবিকভাবে আমাদের মধ্যে প্রাণীর মধ্যে আমরা যাদেরকে দেখতে পাই তাদেরকে প্রাণী সেল দিয়ে তৈরি হবে স্বাভাবিকভাবে, কারণ একটা প্রাণীর মধ্যে গাছের সেল তো কোনোভাবে আসবে না। আর প্লান্ট সেল দিয়ে তৈরি হবে, তার কিছু ইউনিক ক্যারেক্টারিস্টিক্স রয়েছে যেগুলো আমরা পরবর্তীকালে খেয়াল করে থাকবো। তো আমরা ইভলিউশনের দিক থেকে দেখলাম, যেখানে আমরা দু ধরনের সেল দেখলাম, ইউক্যারিওটিক সেল এন্ড প্রোক্যারিওটিক সেল। আর আমরা অর্গানিজমের উপর বেস করে দেখলাম কি দু ধরনের সেল হয়, একটা অ্যানিমাল সেল, একটা প্লান্ট সেল। তো এখানে দুটো স্ট্রাকচার দেখতে পাচ্ছ। এটাকে আমি ইচ্ছা করে একটু ছোট করে এঁকেছি বিকজ আই এম গোয়িং টু ইলাবোরেট দিস স্ট্রাকচার ফার্স্ট এন্ড দেন ইউ ক্যান ইজিলি আন্ডারস্ট্যান্ড দিস স্ট্রাকচার। ওকে, so let me present a story to you, that is in every family there some of us have a big brother. Okay, some of us have a big brother. So usually what usually occurs in the family, that big brother usually commands his small brother. Big brother usually commands his junior ones for the completion of any task. So some of you can understand that if your big brother or sister gets a task from your mother or father, so the task is transferred to junior ones, and the junior ones are bound to do the task. Okay, this is I'm just taking as an example. So in the case of cell, there is typically one big brother and one junior brother. We will be discussing about them. So here the organelle that I have drawn, it is like the head of the cell, or the boss of the cell, who regulates all the functions of the cell. As boss regulates all the functions of an office, all the functions of a house. So the nucleus, so this organelle is named as nucleus. So this organelle is called nucleus, and the nucleus is the boss. Okay, nucleus is the boss of the cell. So what nucleus has? Nucleus has a big brother. That we are taking the example of a family that we have a big brother. So in nucleus there is a part of the cell that is as important as the big brother. So the big brother is the chromatin, or I can call it DNA. Also I can call it chromosome. All these names are same, that is pointing towards the same material, but they have different names in the different phases. So we can call this filament-like structure a chromatin and DNA and chromosome according to the phase we are looking into the cell. So to clarify, this is a chromosome. Okay, this is a structure of typical chromosome. So this is a structure of chromosome, and if I unfold the structure, if I try to unfold the structure, what I will get, so this is the structure what I will get. This structure is called chromosome. This loosely bounded structure is called chromatin, and this structure is called DNA. So this is nothing but DNA, but if I am looking at different stages, then I can see the see the filament in different phases. If the phase is very lightly packed, then it is called DNA because the bases are exposed. So if the DNA molecule is simply packed or simply coiled, I should turn the name coiled. So if they are simply coiled, they are called chromatin, and if they are extensively coiled or extensively condensed in a structure, then this will be called as chromosome. মানে কিছুই না, মানে আমি যদি ডিএনএ টাকে বেশি করে পাকিয়ে রাখি তখন আমি কি পাবো, একটা কয়েল স্ট্রাকচার পাবো। দ্যাট কয়েল স্ট্রাকচার ইজ নাথিং বাট আমার ক্রোমাটিন, এবং এই কয়েল স্ট্রাকচারটা যখন নিজের অ্যাক্সিসের বরাবরে ফোল্ড হবে তখন এরকম একটা শেপ তৈরি করে যার নাম আর কি ক্রোমোসোম। ওকে, so the DNA part, I am referring it as a DNA. So the DNA part of the nucleus is just like a big brother. Okay, so what big brother does, big brother or the DNA sends its message to whom? To his small brother, that is known as RNA. Full form of DNA is deoxyribonucleic acid. Full form of RNA is ribonucleic acid. Okay, I'm not writing the names because the video will be much more longer. I'm just mentioning the full form to you. Later we will be discussing them broadly. So there is DNA, the big brother, and there is RNA, the small brother. The process via which the DNA is converted to RNA, or the message, I prefer the term gene. The process by which the genes of the DNA are encoded in the RNA is called transcription. মানে কি বললাম, মানে ডিএনএ থেকে আরএনএ তৈরি হওয়ার পদ্ধতিটাকে কি বলা হয়? ট্রান্সক্রিপশন। ওকে, and there is a fun fact, that is DNA can itself replicate. The process is called replication. So the copying of DNA into DNA is called replication. The copying of genes from DNA to the younger brother, that is RNA, is called transcription. Okay, and the RNA, the small brother, also goes under a process that is called translation. Also undergoes a process called translation, and via translation the RNA converts into protein or polypeptide. So I have told what is protein, what is polypeptide. Those are discussed in the previous video. So I think it is clear that DNA has some genes. Okay, so the genes are converted into a form that is RNA, and RNA is converted into protein. So what protein will do? Protein will do the functional part. So the functional part of a cell is conducted by protein. So you might be asking that why DNA is converted into RNA, is converted into protein? Why cannot DNA from protein? So the answer is DNA is a huge size of molecule. Okay, and DNA cannot come from the nucleus to the cytoplasm. I will be later naming them, but just to simplify, this huge structure cannot go from here to here. This huge structure cannot pass. So it converts into a smaller part, that is RNA. Okay, so this is how the nucleus works. So nucleus has all the information in it. Information means genes. So what genes are doing? What are genes? So genes are all the information that is required to perform the functions of a cell. Genes are all the informations that is required to do the work of the cell, to maintain the cell, অন্যদের হুকুম চালানোর জন্য এই জিন্সই কাজ করে। বাট এই জিন্স কার মধ্যে থাকে? ডিএনএ। ডিএনএ থেকে ডাইরেক্ট বাইরে আসতে পারে না বিকজ অফ ইট হিউজ সাইজ। So it converts into RNA that is less huge, and the RNA is finally converted into protein, which will be the functional part, মানে যে গিয়ে আর কি কাজ করবে। ঠিক আছে? তো এবার so the nucleus has all the information in it. Nucleus sending his little brother, or the DNA sending his little brother RNA into the cytoplasm. So I have already named this. So there, okay, let me mention this. There are some pores in the nucleus. Those we can be called as nuclear pores. Okay, so nucleus is a double membrane structure, means it has two membrane. One membrane is here, another membrane is here. Here will be a pore that will lead to the outside. Okay, so this is the structure of nucleus. So via nucleus, the RNA part comes to this open area is called cytoplasm. So that this open area is called cytoplasm. What is cytoplasm? Cytoplasm is a thick jelly-like structure that holds all the organelles you can see in this picture. Okay, this is an organelle, this is an organelle, this is organelle, this is the organelle. All the organelles that is present inside a cell is swimming in a jelly-like substance that is called cytoplasm. Okay, cytoplasm consists of several proteins, several lipid molecules, etc., etc. Okay, so the RNA finally has come to the cytoplasm. So I am drawing this as a RNA. This as a RNA. So DNA to RNA that has arrived to the cytoplasm. Now there is another protein I have drawn here. This one's name is ribosome. Ribosome. So this one's name is ribosome. Though it is not this big, this is just like the dot I have drawn here, but still to elaborate the process I have to draw it in big. So there is ribosome. Ribosome is in the cytoplasm, and the RNA is also in the cytoplasm. So RNA is also in the cytoplasm, and there is also ribosome. So I have mentioned the process that RNA is converted into protein via a process called translation. So who does this process of translation? The answer is the ribosomes. The ribosomes of a cell converts the process of translation. So কি হবে? RNA টা আসলো, RNA টা যখন সাইটোপ্লাজমে আসলো, তখন যে সাইটোপ্লাজমের মধ্যে রাইবোসোমটা ছিল সে এসে RNA এর উপর বসবে and সে RNA থেকে প্রোটিন তৈরি করবে। Okay, so there is RNA, this is RNA, and protein and ribosomes will come in it. Ribosomes will come in it and do the job of uncoding, decoding, what you can call, and then give us the final product that is our protein. Okay, so now what is the fate of that protein? এই যে প্রোটিনটা তৈরি হলো, আমি মানলাম এখানে রেপ্লিকেশন হয়ে এখানে একটা প্রোটিন তৈরি হলো। This is the protein, or okay, let me draw it with another color. So this is the protein, this blue dot is the protein. So what the protein will do now? So you have to remember this process that the protein goes into several modifications, because to perform certain actions, the protein that is made in the process of replication is not developed to perform the functions. So just like a Marvel movie, if Tony Stark is going to a war without his suit, so he is not an Iron Man. So this is the protein which is naked, or what you can call, it has not got his armor right now, and to get the armor he is going to these structures, those are called endoplasmic reticulum. I am erasing some part of it. So the protein, to get his armor, goes into these organelles which are called endoplasmic reticulum. Endoplasmic reticulum. Okay. Or I can shortly call them ER. There are two types of ER. You can see what has certain dots in it and one has smooth. One is not have it. So they can be classified as rough, that has the dots in it, and one is called smooth. So which one is rough? This one is rough. Why it is rough? Because it has ribosomes in it. All these dots are ribosomes. So sir, what the ribosomes are doing here? So let me clarify. Some proteins are needed to be expelled from the cell. They need to be go outside of the cell to do their work. So those proteins are tend to come to rough ER for their development. Okay. So what is the function of smooth ER? So it also does the jobs, but it is not associated with the metabolism of protein or the development of protein. It is rather involved in the development of lipids. So rough protein is involvement in the development of protein. Smooth ER is busy in the development of lipids. Okay. So these the protein is developed in the ER. So now the protein got his armor, and what the protein should do? The protein goes a second stage of development in another organelle. That is this organelle I have drawn here, this messy organelle that I have drawn. So this messy organelle is named as Golgi bodies, or you can call it Golgi bodies. You can call them Golgi bodies and Golgi bodies. So how the protein travels from the ER to the Golgi bodies? So answer is very simple. The endoplasmic reticulum, once the proteins is developed, they shift the protein via cargo molecules, or you can call them cargo vesicles. So these are the proteins. Let me rub a bit. So this is the modified protein. And now it will be go on a cargo vesicle. The cargo vesicle will carry the developed protein or the semi-developed protein to the Golgi bodies. And now in Golgi bodies, the protein gets its second sets of armor or the second sets of modifications. Second sets of modifications. So in endoplasmic reticulum, the protein gets first stage of modification, and in the Golgi bodies, the protein goes under second stage of modification. So after the modification protein, the protein exits the cell. I have mentioned that the proteins those are required to go outside of the cell is developed in the rough ER, from rough ER to Golgi bodies, from Golgi bodies to the outside of the cell. So how the protein will go outside of the cell? Via these cargo molecules. So again there will be cargo vesicles. I am not writing the whole thing. Cargo and then secreted, and then the whole protein is secreted. So this is one type of the protein that is required to be secreted, like hormones. Hormones are secreted outside of the cells. So how the hormones are secreted out of the cell? This is the process how the protein goes. The protein first develops in the cytoplasm, then it goes in the endoplasmic reticulum. From endoplasmic reticulum it comes to the Golgi bodies, and from Golgi bodies it is sent outside of the cell. Okay, so this is the process of how a cell works. So there are another sets of proteins. So তাহলে কি সব প্রোটিনই সেলের বাইরে বেরিয়ে যাচ্ছে এরকম তো না। There are some sort of proteins that is required to translocate in the nucleus, that are required to translocate in some of the organelles that I have mentioned here. So those proteins freely develops, means there are also ribosomes that are free in the cytoplasm. মানে কিছু রাইবোজোম আছে যারা কিনা এই এন্ডোপ্লাজমিক রেটিকুলামের মধ্যে বাউন্ড হয়ে রয়েছে, কিছু কিছু রাইবোজোম আছে যারা কিনা সাইটোপ্লাজমের মধ্যে খুলে ফেলে ঘুরে বেড়াচ্ছে। তো যারা সেলের মধ্যে থাকতে চায় তারা কোন রাইবোজোমের কাছে আসবে, যেগুলো ফ্রি অবস্থায় রয়েছে। Those proteins that are required to expel or secreted out from the cells are go to ER, then Golgi, then outside of the cell. Okay, so this is how the endoplasmic reticulum works. This is how the Golgi body works. Okay, now the easy parts of the cell. I hope you have understand. If not, please rewatch this part and I hope you will understand. So now we have mentioned all this. This is Golgi bodies. This is ER. This is nucleus. Okay. So what is this organelle? This organelle is called mitochondria. This organelle is called mitochondria. What is mitochondria? I think everyone should know that mitochondria is called powerhouse of the cell. Powerhouse of the cell. Why it is called the power of the cell? So that is one of your assignments, if possible do, but I can simplify it for you. That is, the mitochondria has some of these like structures, some of these particle like structures, which we called F0 F1 particles. Okay, so these F0 F1 particles forms the energy currency of our body, that is ATP. What is the full form of ATP? ATP is adenosine triphosphate. So how this ATP generates power for our body? There is three phosphates in this ATP. 1 phosphate, 2 phosphate, 1, 2, 3 phosphate, and this is the adenosine. So this, when the phosphate is broken here, it yields very much of energy. That energy is grouped with rather I said coupled with another reactions. So the energy that is yielded due to the breakage of this bond is very high, and the energy is coupled with another reactions. So when I will explain the thermodynamics, I will tell this, and just remember the ATP is responsible for the generation of energy in our body. Okay, so this is mitochondria. Mitochondria is basically providing the power for each and every organelles, those required power to work. যেমন আমাদের খাবার লাগে খেতে, কাজ করার জন্য আমরা যেমন খাবার খাই, তেমনি each of the organelles you are seeing in this picture requires energy, and that energy is fulfilled by this organelle, that is mitochondria. Okay, and there are some other organelles I have drawn. This one is called lysosome. So lysosome has an interesting story. Okay, lysosome. So this is, I think the name is visible. This is lysosome, or I am also mentioning it here. Lysosome. Okay. So lysosome has all the hydrolytic enzymes in it, and it is covered by a lipid membrane. This is a lipid membrane, and all the enzymes, all the hydrolytic enzymes. What enzymes? Hydrolytic enzymes. So all the hydrolytic enzymes are covered inside an organelle, and we call that organelle is lysosome. So what is the function of lysosome? So first let me describe the functions of the hydrolytic enzymes. Hydrolytic enzymes are a set of enzymes that are responsible for degradation of anything. Okay, anything you are seeing inside the cell, that may be DNA. So the enzyme that degrades DNA, যেটা আমাকে DNA নষ্ট করে দেয়, that is DNase. যেটা RNA কে নষ্ট করে দেয়, that is RNase. There are protease, etc. There are many much enzymes in it, but broadly there are DNase, RNase, and protease. What this enzymes does, this degrades the DNA, degrades the RNA, degrades the protein. So this is like a suicide package for the cell. So if the lysosome bursts inside the cell eventually dies. So no, this is not the case. The lysosome is highly maintained, and the process by which a cell undergone death, that is a mechanism called apoptosis. So that is like another lecture. So the hydrolytic enzymes, what it does, it usually protects the cell from outsiders. So you may have heard that some virus is encountering our cell, some bacteria is encountering our cell. So when there is invasion of any of the foreign particles that the cell considers to be dangerous, let me draw it quickly. Suppose this is a cell, and there is something outside, a foreign particle that has somehow entered into a cell. So when the particle enters into the cell, it is also membrane bound. The membrane bound, or the membrane that is covering the foreign particle, have a name called clathrin coated vesicle. So just don't, you may forget the name. So when the foreign particle is entering inside the cell, if the cell decides that it is dangerous for it should have it must eliminate the foreign particle. So what it does, it fuses the lysosome with the foreign particle. Suppose this foreign particle is a virus, and virus usually have RNase or DNase. So when the virus ejects or enters his DNA inside a cell, then the DNA is very much a poisonous particle for the cell. What the cell does is the DNA, which is in a vesicle, and the lysosome, which is also in a vesicle, and these two vesicles fuses with each other, and the fusion leads to the expel of all the enzymes that are inside the lysosome, and eventually degrades the foreign particle. Okay, so if the foreign particle is protein, then the protease will do the job. If the foreign particle is DNA, the DNA will do the job. Okay, so this is the broad function of lysosome, although we will be encountering each of the functions in the future videos. Okay, and there is one organelle that is called centrosome. So what is the function of centrosome? The centrosome, I told you that DNA does replication. So what is the motive of replication? The motive of replication that is one cell differentiates into two cells, and the process goes on. Okay, this process is called mitosis. So when the multiplication process occurs, so also there should be the segregation of DNA. So suppose there is DNA one cell, so there will be duplication of DNA, and this duplicated DNA will be passes to the daughter cells one by one. Here is one, here is one. So the segregation of the DNA are done by the centrosome. Okay, centrosome sort of forms some microtubules which during cell division mainly in the anaphase, in the metaphase helps in segregation of the chromosome. In metaphase, the whole DNA aligns at the mitotic plate, and from the centrosome their microtubules are formed. These microtubules then shrinks, and then these all DNAs are concentrated into the polar region. So this is what the broad functions of the centrosome. Okay, and these lines I have done here, these are nothing but the microtubules. These are the microtubules I have drawn, and the outer part of the membrane is called, the outer part of the cell is called cell membrane. The outer part of the cell is called cell membrane. So there is cell, there is organelles. So everything needs to be packed, okay, in order to keep them shape. So the shape is provided by membrane, and membrane is usually made up of protein, lipid. Okay. So these are all the organelles that a animal cell bears, and in comparison of the animal cell, there is slightly some modification in the plant cell. So there is the outer wall, you can call it outer wall, is called a cell wall. So there is cell wall present in the plant cell. There is no cell wall present in the animal cell. Also there are a organelle called plastid. Plastid is an organelle that is responsible for the photosynthesis. Photosynthesis is the production of glucose by using sunlight and carbon dioxide and water. Okay, so this is the function of plastid. I will be covering all the organelles in detail, and this is a broad gap, or a vesicle you can call, that is called vacuole. So what is a vacuole? Vacuole is an empty space. It may contain food, may contain water, may contain nothing. Okay, so these are the structure of the cell. I think I have cleared all of it. So if any doubt arises, please let me know. Please do comment, share and subscribe with your friends. Thank you.